a frozen traveler from the ancient galaxy, chemistry older than Earth, molecules that shouldn't exist together yet do. And drifting through our solar system right now, a comet that might carry the answer to humanity's oldest question: how does life begin? 3. I-Atlas isn't just breaking the rules of physics anymore. It's revealing something far more profound, something that connects the chemistry of distant stars to the biology of living worlds, something that suggests life's ingredients aren't rare accidents confined to our planet, but universal patterns written into the fabric of galactic evolution. NASA's James Webb Space Telescope has detected compounds in 3I-Atlas that astrobiologists recognize immediately. Prebiotic molecules, the chemical precursors that, under the right conditions, can assemble into the building blocks of life. Not DNA, not proteins, not life itself, but the ingredients that come before, the raw materials from which biology emerges. And these materials didn't form here. They formed around another star in conditions completely unlike our solar system, yet produced the same fundamental chemistry that made life possible on Earth. This changes everything we thought we understood about life's origin. Because if 3I-Atlas carries these molecules, if interstellar comets routinely transport prebiotic compounds between star systems, then life might not emerge independently on each world. It might spread. It might travel. It might be a galactic phenomenon, not a planetary one. Welcome to the story of how one comet from beyond our solar system is forcing us to reconsider whether life on Earth began here or whether it arrived from the stars. November 2025. The James Webb Space Telescope completes another spectroscopic observation of 3I-Atlas. The data downloads to the Space Telescope Science Institute. Researchers open the files, expecting to see the familiar carbon dioxide dominance, the inverted ice ratios, the unidentified absorption features they've been tracking for weeks. What they see instead makes them call colleagues immediately, makes them recheck calibrations, makes them request priority observation time for confirmation, because Webb has detected methanol. Not trace amounts, significant concentrations, and alongside the methanol, formaldehyde signatures and complex organic molecules that indicate carbon chemistry far more sophisticated than simple ices. These aren't exotic compounds unknown to science. They're well-studied molecules. But finding them together at these concentrations in an interstellar comet changes their significance entirely. Methanol and formaldehyde are what astrobiologists call prebiotic molecules, compounds that can participate in chemical reactions leading toward more complex organic chemistry. Not life, not even close to life, but steps on the pathway, ingredients in the recipe, raw materials from which, given the right environment and enough time, biological molecules might eventually emerge. On Earth, these compounds appear in the atmosphere, in volcanic gases, in the chemistry of early oceans. They're part of the chemical landscape that existed before life began. And now, Webb has found them in material that formed around another star, traveled through interstellar space for millions of years, and arrived in our solar system carrying that ancient chemistry intact. Subscribe now, because what this discovery means for understanding life's origins will challenge everything you thought you knew about biology's place in the universe. Let's be precise about what Webb detected and why it matters. Methanol, chemical formula CH3OH, a simple alcohol consisting of one carbon atom, one oxygen atom, and 4 hydrogen atoms. In terrestrial chemistry, it appears in wood decomposition, volcanic emissions, and atmospheric reactions. In prebiotic chemistry, it serves as a carbon source for more complex molecule synthesis. Formaldehyde, chemical formula CH2O, even simpler. 1 carbon, 2 hydrogen, 1 oxygen. Toxic to living organisms in high concentrations, but essential in the chemistry that comes before life. Formaldehyde can polymerize, linking molecules together into longer chains. Those chains can become sugars. Those sugars can become ribose. and ribose is the sugar backbone of RNA, one of life's fundamental molecules. These aren't hypothetical connections. Laboratory experiments have demonstrated these reaction pathways. Mix formaldehyde and other simple molecules under conditions resembling early Earth, add energy through ultraviolet light or electrical discharge, and complex organic compounds begin forming. Amino acids, nucleobases, the chemical building blocks that eventually assemble into proteins and genetic material. But here's what makes the 3I Atlas detection extraordinary. These molecules didn't form on Earth. They formed in another stellar system under completely different conditions, around a different star with different chemistry, different temperature, different radiation environment. Yet they produce the same prebiotic molecules. This suggests something profound, that the chemistry leading toward life isn't unique to our solar system's particular conditions. It's a universal pattern, a natural outcome of carbon chemistry occurring anywhere in the galaxy where the right basic ingredients combine. Carbon, oxygen, hydrogen, nitrogen, the most abundant reactive elements in the universe. Put them together under conditions where molecules can form, and you get methanol, formaldehyde, and other organic compounds. Not because Earth is special, because chemistry is universal. Understanding where these molecules came from requires understanding where 3I Atlas itself originated. Orbital mechanics provides clues. The comet's trajectory, extrapolated backward through time, doesn't point toward any nearby star. It comes from the general direction of the galactic thick disk, that region of older stars orbiting above and below the galaxy's main plane. Spectroscopic analysis adds detail. The carbon dioxide to water ratio, the methanol concentrations, the metallic traces Webb has detected, including nickel and iron, all suggest formation in an extremely cold environment, colder than the outer regions of our solar system. Temperatures where carbon dioxide freezes more readily than water, where complex carbon compounds can condense from gas directly into solid ices. Red dwarf stars create such environments. Small stars, barely 20 to 40% of our sun's mass, producing a fraction of its luminosity. Around these dim stars, the region where ices condense lies much closer to the star than in systems like ours. The protoplanetary disks surrounding red dwarfs are cold, carbon-rich, and produce comets with chemistry very different from solar system objects. If 3I Atlas formed around a red dwarf, it could be billions of years old. Red dwarfs burn slowly, living for trillions of years. Their planetary systems stabilize early, and their comets, once ejected into interstellar space through gravitational interactions, wander the galaxy carrying preserved samples of ancient stellar chemistry. That's what makes this comet so valuable. It's not just showing us the chemistry of another star system. It's showing us chemistry from an earlier era of galactic evolution. From a time when the Milky Way was younger, when stellar populations were different, when the chemical composition of the interstellar medium hadn't yet been enriched by as many generations of supernovae. 3I Atlas is a time capsule, a chemical archive, a frozen library of molecules preserved across billions of years and delivered to our neighborhood by chance. The discovery of prebiotic molecules in 3I Atlas resurrects one of astrobiology's most controversial yet compelling ideas, panspermia. The hypothesis, proposed in various forms over centuries, suggests that life doesn't necessarily originate independently on each world. Instead, the ingredients of life, or perhaps life itself in dormant forms, travels between star systems on objects like comets and asteroids. Traditional panspermia faced major objections. How could fragile organic molecules survive the harsh conditions of interstellar space? Cosmic radiation should destroy them. Ultraviolet light should break their bonds. The extreme cold should alter their structures, but 3I Atlas demonstrates survival is possible. The methanol and formaldehyde Webb detected formed millions of years ago around another star. Those molecules traveled through interstellar space exposed to cosmic radiation, bombarded by high-energy particles, subjected to extreme temperature variations, and they survived, intact, chemically active, ready to participate in reactions when delivered to a new environment. This changes the panspermia hypothesis from speculation to testable science. We're not asking whether organic molecules could survive interstellar travel. We're observing that they do. The question becomes, how common is this? How many interstellar comets carry similar chemistry? And what happens when they deliver those molecules to young planets? Comment below. If life's ingredients regularly travel between stars, does that make life more common or more connected than we imagined? Think about the implications for how life begins on any world. Traditional origin of life models assume chemistry starts from scratch on each planet. Simple molecules in the atmosphere or oceans, energized by lightning or ultraviolet radiation or volcanic heat, gradually react to form more complex compounds. Over millions of years, this chemistry becomes sophisticated enough that self-replicating molecules emerge. Then Darwinian evolution takes over, and life diversifies. This process requires time, lots of time, and specific conditions, the right temperature range, liquid water, energy sources, protection from too much radiation but enough to drive reactions, a delicate balance. But what if planets don't start from scratch? What if they're seeded with prebiotic molecules delivered by impacting comets? Earth's early bombardment phase, 4 billion years ago, involved countless comet and asteroid impacts. We've assumed those impacts delivered water, which is crucial for life. But if interstellar comets regularly pass through the inner solar system, if some of them impact young planets, then they're also delivering complex organic molecules, not just water, not just simple gases, actual prebiotic compounds that have already undergone millions of years of chemistry in stellar environments before arriving. This jumpstarts the process. Instead of beginning with the simplest possible molecules and working up to complex organics over eons, young planets receive a chemical head start. They inherit molecular libraries from older stellar systems. They benefit from chemistry that occurred elsewhere. And if this happens commonly, if interstellar comets regularly distribute organic molecules throughout the galaxy, then every young planetary system receives these chemical gifts. Every potentially habitable world gets seeded with the same fundamental ingredients. Webb spectroscopy revealed another crucial detail about 3-I-Atlas chemistry: metallic traces, nickel and iron signatures appearing in the spectrum alongside the organic molecules. These metals aren't present in large quantities. They're trace elements. But their presence tells a story about the environment where 3-I-Atlas formed. In protoplanetary disks, metals don't remain in gaseous form. They condense into solid grains at high temperatures close to the star. Those grains then get incorporated into larger bodies, asteroids, and rocky planets in the inner regions of planetary systems. But in the cold outer reaches where comets form, metals shouldn't be abundant. The temperatures are too low for metal-bearing compounds to condense efficiently. Comets should be mostly ices with minimal metallic content. Yet 3-I-Atlas shows metal signatures. This suggests one of two possibilities. Possibility 1. It formed in a protoplanetary disk with unusual chemistry, perhaps around a metal-rich star, or in conditions where turbulence mixed materials from inner and outer regions more thoroughly than in our solar system. Possibility 2. It incorporated material from interstellar dust during its millions of years traveling between stars. Tiny grains containing metals, produced by supernovae and planetary nebulae, could have accumulated on the comet's surface as it drifted through molecular clouds. Either way, the metals add complexity to 3-I-Atlas chemical profile. They indicate that this object experienced a history unlike typical solar system comets. It sampled different environments. It incorporated materials from multiple sources. It's a composite archive of galactic chemistry. And those metals might be relevant to prebiotic chemistry. Iron, in particular, serves as a catalyst for organic reactions. In early Earth chemistry, iron-sulfur minerals at hydrothermal vents may have catalyzed reactions leading toward metabolism. If interstellar comets deliver both organic molecules and catalytic metals, they're providing not just ingredients, but also the tools for chemical evolution. The Europa Clipper encounter takes on new significance in light of these chemical discoveries. The spacecraft isn't just attempting to sample interstellar dust. It might be collecting prebiotic molecules from another star system. If SUDA, the surface dust analyzer, captures particles from 3 I-Atlas, if those particles contain the methanol and formaldehyde that Webb detected, then laboratory analysis back on Earth could reveal details invisible to telescopic observation. Isotopic ratios in the carbon, oxygen, and hydrogen atoms would pinpoint the formation environment more precisely. Different stellar environments produce different isotopic signatures. Measuring those ratios in actual samples, not just through spectroscopy, provides hard constraints on where 3 I-Atlas formed. Molecular chirality might be detectable. Many organic molecules can exist in mirror image forms, like left and right hands. Terrestrial life uses specific chiralities, left-handed amino acids and right-handed sugars. If 3 I-Atlas molecules show chirality preferences, it could indicate photochemical processes during formation or suggest whether prebiotic chemistry naturally favors certain orientations. Trace compounds too faint for Webb to detect might appear in particle analysis. Complex organics present in tiny concentrations, nitrogen-bearing molecules, sulfur compounds, each one adding detail to the chemical inventory this comet carries. This is why the Europa Clipper encounter matters. Remote sensing with telescopes reveals overall composition. Direct sampling reveals molecular-level detail, and that detail could transform our understanding of prebiotic chemistry from theory supported by laboratory experiments to theory confirmed by actual interstellar samples. 3 interstellar objects have now been detected and studied. Each one teaches different lessons about galactic chemistry and life's potential distribution. 1. Oumuamua. The first, the mystery, no detectable chemistry, no cometary activity, but non-gravitational acceleration suggesting something was outgassing invisibly. We learned that interstellar objects can be compositionally diverse, possibly including types we haven't categorized. 2I Borisov, the conventional one, water-dominated chemistry, matching solar system comets. Predictable behavior. This taught us that at least some interstellar comets form through processes similar to local ones, that our solar system's chemistry isn't unique. 3 I-Atlas, the game changer, carbon-rich chemistry, prebiotic molecules, behavior-violating thermal models. This is teaching us that other stellar environments produce not just different comets, but potentially significant sources of complex organic chemistry. 3 visitors, 3 completely different chemical profiles. What does this distribution tell us? That the galaxy contains incredible chemical diversity, that different stellar environments produce different outcomes. That comets aren't just frozen water and dust, but mobile chemical laboratories transporting materials between star systems. And if 3 I-Atlas isn't anomalous, if other carbon-rich, organic-bearing interstellar comets regularly pass through planetary systems, then every habitable world throughout the galaxy receives these chemical deliveries. The ingredients of life aren't confined to individual stellar systems. They're galactic resources, distributed by gravitational dynamics and collected by any planet fortunate enough to be in the path. The Vera Rubin Observatory, scheduled to begin operation soon, will revolutionize interstellar object detection. Its wide-field telescope will scan the entire visible sky every few nights, identifying faint moving objects that previous surveys missed. Astronomers predict Rubin will detect dozens to hundreds of interstellar visitors per year, not just the bright ones that current surveys catch. Smaller objects, darker objects, objects passing through the outer solar system that would otherwise go unnoticed. Each detection is an opportunity to study the chemistry of different stellar environments. Each interstellar comet carries a unique chemical signature from its formation location. By building a statistical sample, by analyzing dozens of these visitors, we can map the chemical diversity of our galactic neighborhood. Some will be water-rich, like Borisov. Some will be carbon-rich, like 3I Atlas. Some might show chemistry we haven't imagined yet, formed in stellar environments unlike any we've studied. And among those dozens of visitors, how many will carry prebiotic molecules? How many will show organic chemistry complex enough to be relevant for life's origin? If 3I Atlas isn't rare, if organic-bearing interstellar comets are common, then the Vera-Rubin era will prove that prebiotic chemistry is continuously distributed throughout the galaxy. This has profound implications for astrobiology. It suggests that habitable planets don't develop their chemistry in isolation. They're embedded in a galactic ecosystem where materials flow between star systems, where every world receives chemical contributions from beyond its local environment. Step back from 3I Atlas specifically. Consider what this discovery suggests about life in the universe. We've always framed the question of life's origins as what special conditions must exist on a planet for life to begin? What's the recipe? What's the sequence of events? How rare or common are those conditions? But if interstellar comets routinely deliver prebiotic molecules, the question changes. It becomes, given that organic chemistry is continuously supplied to every planetary system, what conditions prevent life from starting? The universe might not be asking how life begins. It might be constantly trying to start life everywhere. And the interesting question is, where does it succeed versus where do conditions prevent it? This is a fundamental reframing from life as rare accident requiring perfect conditions to life as natural outcome that emerges wherever those conditions aren't actively hostile. The chemistry 3I Atlas carries isn't exotic or unusual. It's universal chemistry, carbon, oxygen, hydrogen, nitrogen combining into molecules that can react further. This happens in stellar atmospheres, in protoplanetary disks, in molecular clouds. Anywhere these elements exist in conditions where molecules form. And once formed, those molecules travel. Ejected from planetary systems, drifting through interstellar space, falling onto other worlds, continuously redistributing chemistry throughout the galaxy, life on Earth might not have originated purely from terrestrial chemistry. It might have been influenced by, seeded by, or jump-started by organic molecules delivered from interstellar sources. Not living organisms, not DNA from space, but the chemical ingredients that allowed biology to emerge faster or more readily than if Earth had started with only the simplest possible molecules. As I record this, 3I Atlas continues its journey outward. The methanol and formaldehyde Webb detected remain frozen in its nucleus. The prebiotic chemistry it carries will eventually be lost when the comet fades beyond detection, unless Europa Clipper succeeds in collecting samples during its passage through the dust trail. But even if we lose 3I Atlas, even if no samples are collected, this discovery has already transformed how we think about life's place in the universe. For decades, astrobiology asked, are we alone? Is life rare? Did Earth get lucky with the perfect conditions? 3I Atlas suggests different questions. How connected is life? How much does chemistry flow between star systems? Are we isolated accidents or participants in galactic scale distribution of life's ingredients? The molecules in this comet aren't alive. They aren't even close to alive. But they're the starting point, the chemical alphabet from which biology's language eventually emerges. And finding that alphabet written in material from another star system, formed under completely different conditions, yet producing the same fundamental chemistry, suggests that life's ingredients are universal. Not life itself, not yet. But the potential for life, the chemical foundation, the raw materials from which, given time and suitable environments, biology can emerge. If 3I Atlas represents typical chemistry for carbon-rich interstellar comets, if the Vera Rubin Observatory confirms that organic-bearing visitors regularly pass through our detection network, then every planetary system in the galaxy receives these deliveries. Every young Earth, every potentially habitable world, every moon with subsurface oceans, all of them collecting organic molecules from interstellar sources, building chemical libraries that include contributions from dozens or hundreds of different stellar environments. This doesn't prove panspermia. It doesn't prove life spreads between stars. But it proves that chemistry does, that the ingredients of life travel, that no world is chemically isolated, that the universe continuously tries to supply habitable planets with the molecules they need to get biology started. Comment below. Does this discovery make you think differently about life's origins on Earth? And subscribe, because the data from 3I Atlas will be analyzed for years, and as more interstellar visitors are detected, we'll learn whether organic chemistry is rare or whether it's one of the galaxy's most common gifts. 3I Atlas came from the stars carrying molecules that might explain how life begins, not just on Earth, potentially everywhere. Because if chemistry is universal, if prebiotic molecules travel between stellar systems, if every world receives these chemical seeds, then maybe life isn't the exception. Maybe life is what chemistry does when given enough time and the right conditions. And maybe 3I Atlas, traveling from an ancient red dwarf in the galactic thick disk, carrying methanol and formaldehyde frozen for millions of years, is showing us that those conditions aren't rare accidents confined to one special planet. They're written into the chemistry of the galaxy itself. Subscribe for more real science, verified data, and the mystery still unfolding above us.